The NEON Ambassador Program connects a cohort of researchers and educators eager to enhance NEON's engagement and reach a wide range of scientific and applied research communities. The program is designed to equip ambassadors to better use NEON data, samples, or infrastructure to accelerate scientific discovery, scale our educational opportunities, increase participation of the scientific community, and inform public policy at a continental scale. Our current cohort of ambassadors, which is focused on early career scientists, will commence their second year of the program this October. These researchers will actively participate in NEON's upcoming Convergence Summit and work in their second year to plan and implement a capstone project with advice and guidance from their assigned mentors. NEON is excited to maintain this program moving forward and will be assessing options for the next cohort and a related timeline early next year following the Convergence Summit this fall. We encourage those of you who may be interested in the Ambassador Program to look for additional updates regarding the application process through NEON's communication channels early next year. So like I said, today we have 4 NEON ambassadors out of the 7 in the current cohort that will give us a presentation. Each one of these folks will give a short talk, and then at the end of that talk, I think we will have the opportunity to take one question from the Q&A if there is one ready to go. But if not, we'll move to the next speaker and hold a general Q&A at the end with all speakers combined. So do add your questions for each talk to the Q&A as you think of them and as they're coming up. All right, I'm very happy to introduce our first NEON ambassador who will present to us today, Reid Longley. Reid is an assistant professor at NC State University in the Department of Crop and Soil Sciences. Reid completed his PhD at Michigan State University, followed by a postdoc appointment as a director's postdoctoral fellow at Los Alamos National Lab. Reid's research uses metagenomics and transcriptomics to study interactions between microbes and how these interactions impact plants in agricultural and natural systems. Take it away, Reid.
All right, thank you so much. Okay, so my name is Reid Longley, like was said. I'm an assistant professor currently at NC State University. And today I'll be talking about some of my work that was largely done at Los Alamos, but that we're continuing here, looking into the detection of ultra-small prokaryotes in both New Mexican soils and public metagenomic sequencing data. So just a quick about me. I got started in my undergraduate research in Dr. Scott Miller's lab at the University of Montana, where I studied the evolution of thermotolerance in hot spring cyanobacteria. And then I went on to do my PhD at Michigan State University under the guidance of Greg Bonito, where I looked at interactions between environments and their microbiomes in systems like crops, as well as corals and mushrooms. Following this, I had a postdoctoral appointment at Los Alamos National Labs in Dr. Patrick Chain's group, looking at the fungal microbiome and the evolution of bacterial-fungal interactions. And now I'm an assistant professor, just started in July at NC State University, and I'm interested in soil and plant microbiomes as well as interactions between microbes. So before I get into the prokaryotic portion of this, I just wanted to give a little bit of background on the study where this data is coming from. So this was a collaboration with Magoon Shakya at Los Alamos. And initially we were interested in studying viruses from 3 unique sites in New Mexico. We were trying to discover some novel viruses and thought that New Mexico is undersampled and these high elevation communities are likely a unique source of viruses that hasn't been thoroughly assessed. So to assess these viral communities, we use 2 methods. One is bulk metagenome sequencing that maybe most of us are familiar with, where you just take all of the DNA available in a community and sequence it. So this would pick up everything there. And then the second method is using size filtration, where we pass these samples containing intact cells through increasingly smaller filters in order to enrich for viruses while removing some of these larger microbes. And this creates more of a metavirome. And what we found is, as expected from some previous work done on soil viral communities, that viral counts were consistently enriched by this filtration. As you can see here in the yellow, the metaviromes consistently had substantially higher numbers of viruses recovered. And in this Venn diagram here, we can see that the number of unique viruses in metaviromes was also much higher compared to the metagenome sequencing method. But while processing this data, we also started to find that maybe somewhat unexpectedly, there was a large number of bacteria that had passed through even these smallest filters. And we found that these bacteria were from a unique group called the Pottesi bacteria. This is a superphylum of bacteria that is also known as the candidate phyloradiation. It's a very unique group of bacteria. Only a few representatives have ever been cultured, and none have been cultured from soil. But some metagenomes and amplicon sequencing efforts appear to have an enrichment of these taxa. And so some background on these. So the Pottesi bacteria and another group of archaea called the nanoarchaea are really known as these ultra small prokaryotes. So that's why they're able to pass through these filters that I discussed before. And ultra small prokaryotes are a very unique group of organisms, and they're thought to primarily live on the surface of other cells, as you can see in this representation here, that they're living and acting as parasites on other bacteria potentially. And like I said, they're primarily associated with this Pottesi bacteria superphylum and the nanoarchaea phylum. There are some estimates that in certain environments, especially ground and wastewater, that these might represent up to 15% of total prokaryotic diversity. So a large swath of pretty unknown bacteria and archaea are in this group. And it is difficult to assess these in soil because of this extreme diversity. But one way to do it, as we show, is that size filtration might help increase the recovery of these unique small cells. And this is exactly what we found. Looking back at the same samples that I showed for the metavirome studies, we found that in filtered, shown in purple here, we found that there was a much higher recovery of these Pottesi bacteria metagenome assembled genomes, or MAGs, representing up to 47% of the MAGs recovered from the metavirome. And as many as 38 were recovered from a single sample. While on the other hand, these bulk metagenomes, where you might have sequencing getting overwhelmed by other taxa, we found that only 7% of the MAGs recovered from metagenomes were from this unique group. And so we ended up recovering a total of 489 MAGs from this Pottesi bacteria group. But we were interested to see if this is a common occurrence, and we were interested in assessing this beyond our samples. And so we went to public data and found a group of SRA experiments. So where sequences had been uploaded, we were able to find experiments where we knew that data had been processed similarly, that the size filtration had occurred for the cells, but only viruses had been assessed and the bacteria were not directly assessed fully. And between the New Mexico soils and the public data that we've assessed, we have thus far recovered 945 Pottesi bacteria MAGs. So out of all of these metaviromes, that represents about 38% of the total recovery. And in this public data, we also found an enrichment of these nanoarchaea that I mentioned, recovering 294 MAGs from this unique nanoarchaea group from samples taken across the country, as well as a sampling done in Switzerland that's not shown here. And I won't get too deep into this just to keep ourselves on time, but what we found is this extreme diversity. So each point on this plot on the right represents a single MAG. And in yellow and green are our ultra small prokaryotes, so the ultra small archaea and bacteria. And in purple are all other types of prokaryotes. So you can see that these have some pretty unique genomes. They're extremely small. So you can see on this genome size plot, they're usually smaller than 1,000,000 base pairs, which for bacteria and archaea is quite small. And they generally have lower GC content, which generally means that there's less coding content in the genome, that they might be losing some non-essential functions as they start to rely on their host. So you can really see how unique these genomes are here. And then looking at this phylogeny, again, I won't go too much into this, but we recovered an extremely high diversity of these groups. This inner ring is showing kingdoms, so the green is representing archaea, and again, this group is primarily the nanoarchaea, while this orange is representing all bacteria. But then if we look at this ring in the middle, the green is representing Potassibacteria, whereas all other colors are representing different phyla of bacteria. So you can see that this kind of greenish color in the second ring is really taking up most of that phylogeny, indicating that we've recovered a lot of these ultra small bacteria based on these 16S signatures. So in conclusion, I think we've shown that, and others have shown before, that soils are known to host this rich diversity of very unique and ultra small prokaryotes. And these can be studied by using some similar methods to how virome-enriched sequencing is generally performed. This is an effective way to recover these ultra small taxa. And like I showed on the previous slide, ultra small taxa appear to have these unique genomes. So a lot of our upcoming work is to try to explore some of these genomes a little bit more. Particularly, I'm looking into seeing if we can try to identify some hosts, some bacterial hosts of these tiny bacteria, in order to look for horizontal gene transfer signatures that might indicate a close relationship. We're also interested in trying to develop novel methods to potentially co-culture these things from soil. And then I'm working on currently using some other publicly available metagenomes from sources like NEON and JGI to look for these in either size filtered or non-size filtered metagenomes to try to recover further diversity. And with that, I am happy to take any questions if we have time.
Oh, thanks. Fascinating. I'm not seeing anything in the Q&A yet, but does anyone will just maybe give it 10 seconds, see if any questions, or if anyone wants to raise their hand, we can give you speaking permissions if there's a quick question for Reid. All right, well, let's keep going. I think that was really interesting, and I think the questions will percolate. Apologies as we go on to our next speaker. Let's do that. Our next speaker is Alexi Besser. Alexi is a postdoctoral research scholar in the School of Earth and Space Exploration at Arizona State University, where she is currently working on a collaborative NSF macrosystems biology and NEON-enabled science grant studying plant litter decomposition across drylands. Alexi received her PhD in biology from the University of New Mexico. As a broadly trained ecologist, Alexi integrates molecular, isotopic, and genetic techniques to investigate the biosynthesis and movement of macromolecules within and among organisms across a variety of systems and scales. Thanks, Alexi. Take it away.
Okay, awesome. Thank you so much for that introduction. I'll get my screen shared here. Okay, cool. So today I'll be talking to you all about a study I started in my first postdoctoral appointment at ASU working with Elizabeth Trembath-Reich. So we know that microbes are everywhere, and we're getting a better understanding of their ubiquity in Earth's atmosphere, where they can reach concentrations of hundreds of thousands to millions of cells per cubic meter of air. Microbial ecologists have long known that the atmosphere is an important transport medium for moving microbes between habitats, helping seed new environments, maintain biodiversity, facilitate gene flow, and even disperse pathogens. And as you can see in this figure, bacteria and archaea, aerosolized from terrestrial habitats, can be transported thousands of kilometers across the ocean, which really highlights the importance of the atmosphere in influencing microbial ecology globally. But what happens while microbes are in the air? Are they simply undergoing passive transport, or might they be active in interacting with their environment? So I am part of an international team of researchers funded by the Human Frontier Science Program, studying the atmosphere as a potential microbial ecosystem, where I focus primarily on understanding the composition of microbial communities in the near-surface atmosphere, with the goal of answering this first question here. Does the atmosphere host resident microbial communities? So when we were thinking about the conditions that might influence microbial dispersal into and survival in the atmosphere, we realized there are certain atmospheric conditions like temperature, incoming solar radiation, relative humidity, and even wind speed and direction that follow predictable patterns over the course of a day, particularly in the desert. So we asked, does the composition and diversity of the aeromicrobiome vary over Diel timescales too? And we investigated this at the Santa Rita Experimental Range NEON site in the Sonoran Desert south of Tucson, Arizona. We sampled near-surface atmospheric microbial cells by filtering air through a polypropylene electret microfiber filter at 300 liters per minute for 4 hours. We minimized contamination by using extensive sterilization procedures for our equipment and taking preventative measures like wearing Tyvek suits and masks to reduce the potential collection of our own cells in microbiomes. Sampling at a NEON site was ideal for this project because we could download a ton of metadata from the nearby Flux Tower to compare with our own data. And so I just want to put a quick plug in for NEON's research support services, which really made all of this work possible. So we selected 3 different time windows for our study, which encompass the greatest variability in the atmospheric conditions I showed earlier. Our first time window from about 2:20 to 6:20 a.m. had the lowest temperatures, highest relative humidity, and gentle winds from the southeast. Our second time window from 7 to 11 a.m. represented a transitionary time frame with increasing temperatures, decreasing relative humidity, and gentle winds from the west. And then our last time window from about 12:30 to 4:30 p.m. had the highest temperatures, lowest relative humidity, and slightly stronger winds from the northwest. We extracted genomic DNA from our filters using a modified CTAB method, and then sent them to Rush University Genomics and Microbiome Core Facility for 16S rRNA gene and ITS sequencing, following the same procedures used by NEON for their microbe marker gene sequencing samples. So looking at our 16S rRNA gene sequencing results first, we found that the diversity of bacteria and archaea present in the air was greatest during that transition time window, between about 7 and 11 a.m. And we also see that each time window had distinct bacterial and archaeal assemblages, as seen in this plot of the relative abundances of phyla. Our 2:20 to 6:20 a.m. time window had greater relative abundances of Bacillota, while our 12:30 to 4:30 p.m. time window had greater relative abundances of Pseudomonadota. Across samples, there were also a few amplicon sequence variants, which can be thought of as similar to species, that were particularly abundant, including one for Geodermatophilus obscurus, which we actually have an approximately 30% complete metagenome assembled genome for, which isn't that high, but I'll talk more about that later. And then we also had an ASV in Selenococcus rosus, and then one in Paracoccus, again, that were quite abundant across samples. But to better visualize variation in the bacterial and archaeal assemblage composition across our time windows, we use non-metric multidimensional scaling, so NMDS, of Bray-Curtis dissimilarities. So in this plot, samples that plot closer together have more similar communities, and samples that plot farther apart are more distinct. And we see that mean relative humidity and mean temperature strongly influence differences among time windows in this plot, really driving that variation among NMDS1 axis. And then things like mean shortwave radiation, mean wind direction, and mean wind speed might be driving variation within time windows, a little bit more on that NMDS2 axis. So we then used indicator species analysis to investigate which ASVs drove differences in bacterial and archaeal assemblage composition among our time windows. So in this plot, each row along the y-axis represents one sample, and then we have significant ASVs on the x-axis. The size of the circle represents the relative abundance of that ASV in that sample, and then the color just indicates the phylum. So if there's no circle, that means that ASV was not found in that sample. And according to this analysis, we found that the 7 to 11 a.m. time window had the most unique ASVs, while that 12:30 to 4:30 p.m. time window had no completely unique ASVs. Now, looking at fungi using ITS sequencing, we found that the diversity of fungi present in the air was actually greatest during that early morning time window between about 2:20 and 6:20 a.m. And again, that we see some differences in the relative abundances of fungal orders across time windows. And interestingly, there were 2 ASVs that were highly abundant across samples here. Cladosporium cladosporiotes, which is a very common darkly pigmented mold, very highly abundant. And then also a species of Alternaria, which is a saprophyte and an opportunistic plant pathogen. And then looking at our NMDS plot here now, again, we see some distinctions in fungal assemblages across time windows. However, temperature here was only marginally significant as a driver, with a p-value slightly above 0.05. So we aren't quite sure what's driving these differences yet. And according to our indicator species analysis, each time window had several completely unique fungal amplicon sequence variants. And we also found that the 2:20 to 6:20 a.m. and 7 to 11 a.m. time windows shared a few more ASVs in common that were still distinct from that 12:30 to 4:30 p.m. time window. Okay, but where are these microbes coming from? So we are also working on analyzing some soil microbe marker gene sequence samples collected by NEON in the same month as our air samples. So in this figure here, there are about 5 plots, soil plots that are near the flux tower where we sampled our air samples. And that's going to be the Santa Rita 004 and then Santa Rita 043 through 053 plots. And they have a distinct bacterial and archaeal assemblage composition compared to the other soil plots because the soils are quite different. They're different types and different ages. But just comparing the relative abundance plots between the soil and our air samples, we can spot some similarities and differences between the two environments. I initially analyzed these two data sets separately, so I'm working on merging analysis pipelines and ASV tables so that we can more directly compare them on the same NMDS plot, and then also use other statistical analyses like source tracker to estimate proportional contributions of local soils to the aeromicrobiome. And I'm especially curious to try this and make these more direct comparisons between soil and atmospheric fungal assemblages, because as you can see in these relative abundance plots here, they look almost nothing alike. The colors don't really match up. And then lastly, as I kind of mentioned earlier, we performed ultra deep metagenomic sequencing for one pooled genomic DNA sample per time window. Elizabeth is helping analyze this data, and right now we're still trying to make sure we can pull out bacterial and archaeal contigs from what are likely a lot of unclassified and complicated eukaryotic contigs. But initial analysis comparing our relative abundances of bacterial and archaeal phyla from our amplicon sequences, which I combined into one bar in this plot per time window, and then those from our metagenomic sequences, which we assessed using the microbial fraction function from the single package, are really exciting. We see that there are some similar patterns, but it looks like our metagenomic sequencing picked up on 2 phyla that we saw in soil, but that we aren't seeing in our amplicon sequences. So there's a lot more to come out on this later as we still work through it. And with that, I just want to say thank you very much to Elizabeth, my first postdoc advisor, and then Heather Troop, who's my current postdoc advisor, and then Rachel Lappin, my colleague at Moash, who really helped develop a lot of the filtration methods for this project, and then all the PIs on the Human Frontier Science Program grant, James Bradley, Jackie Gordiel, and then Chris Greening, and everyone else listed here. So thank you.
Another fantastic talk. Thank you, Alexi. That is super interesting. Similar to Reid, I just wanted to kind of give a chance if there was a question, and it does look like we had one. So did you use primers in the amplicon sequencing? And if so, how did you determine which ones?
Yeah, so we used primers. We chose the same primers that NEON uses because we wanted to make sure that our data was directly comparable to the NEON data set. So it was 515FY and 926R for the 16S, and I think like ITS1 and ITS2 for the ITS.
Great. Well, thank you so much, Alexi. And other questions, if you think of them related to Alexi's talk, just pop those in the Q&A and we'll definitely have more time at the end for discussion. All right, let's get ready for our next ambassador. That's going to be Yujie Liu. Yujie is a postdoctoral scientist at the Center for Ecosystem Science and Society at Northern Arizona University. She is working on an NSF-funded project about evaluating the continuity of NEON and AmeriFlux data streams. Her recent interests also include applying machine learning and process-based models to better understand how cold season climate influences soil carbon dioxide fluxes. So excited for your talk. Thanks, Yujie.
Yes, thank you for the introduction. Let me share my screen. I'll be talking about our findings for the continuity of NEON and AmeriFlux data streams from collocated sites. I will start with some background for the project. You may already know that AmeriFlux network has been connecting independently managed sites in diverse ecosystems. Some AmeriFlux data records are more than 25 years. Here I'm showing an example on the left side. Howard Forest is the longest running tower site in the world since 1991. Most AmeriFlux sites operate 3 to 5 year time horizon. It's all depending on the fundings. However, none are guaranteed to continue for decades into the future. On the other hand, NEON was initiated in 2017, aiming to provide high-quality data over a 30-year period. Combining new NEON data with pre-existing AmeriFlux data could potentially provide robust understanding of the past and future trajectory of ecosystem change. This brings us to a question: can we directly stitch together the two data streams? Before that, we need to understand the level of continuity between the two data streams in terms of meteorology, fluxes, and the flux footprints. To answer the question, we identified 12 pairs across 8 NEON domains. There are 2 study sites, dryland sites in Arizona and New Mexico, 1 subtropical forest in Florida, 2 forest sites in New England region, 1 study site in Great Lake area, and 1 prairie site on Great Plains, 1 forest site and 1 tundra site in Rocky Mountain regions, and 3 paired sites in Alaska. So now we come to the results about half-hourly CO2 fluxes. In the scatter plots, I put data from AmeriFlux on x-axis and data from NEON on y-axis. The first thing to notice is that there was only 4 sites with good agreements. You can tell from the scatter plots, the points form tight ellipses closely following the one-to-one line. But you need to notice even for the 4 sites with good agreement, the intercept was not 0 and the slope was not equal to 1, indicating systematic offset between AmeriFlux and NEON measurements. For all other paired sites, we observed weak agreement. The scatter plots look like diffuse point cloud with high variability and noise. You can also notice substantial deviation from the one-to-one line. Now we need to ask an important question: to what extent offsets or disagreement observed at half-hourly scale propagate to the annual scale? To ensure valid calculation for the annual sums, we first gap-filled the half-hourly data using a machine learning model called XGBoost. For more information about gap filling, you can check out our paper from earlier this year. Now we are looking at the results for the annual sums across different sites. An important message is that the annual CO2 fluxes integrals provide contrasting views of carbon balance between NEON and AmeriFlux. Even for the sites, the 4 sites, we observed good agreement at half-hourly scale for Berlite, Haley, Harvard, and UD. They also don't show good agreement in annual sums. If you take a close look at the results for Barrow and UD, you might notice the similar wiggles of the annual variations. That means they have almost a consistent offset of the values for annual sums between AmeriFlux and NEON, but you don't spot it anywhere else. To understand what is happening with the differences with measurements from NEON and AmeriFlux, let's figure out what the tower says now. Flux footprints are all about where the measured fluxes are coming from surrounding landscapes. There are a couple of points I want to make here. First, the fact is for each paired sites, the measurement height of NEON was consistently higher than that for AmeriFlux. As a result, the footprint area from NEON would be larger than that for AmeriFlux. But only for 2 out of the 12 total paired sites, we found overlapping of the footprints that's Bartlett and Harvard Forest. For other sites, for example, I'm showing the results for Naval Ridge, the tundra pairs, the two towers, they are located more apart, so the footprints are not overlapping. Based on the information of flux footprints, we can further explore the differences in land cover and EVI. We computed dissimilarity matrix with 0% means identical composition and 100% means complete dissimilarity. We plotted the dissimilarity of land cover x-axis and the dissimilarity for EVI on y-axis. The first thing to notice is that there are a couple of sites with similar land cover and EVI. And Howard, Bartlett Forest, and Haley, they are popping up again here. This is consistent with what we saw from the sites with good agreement at half-hourly scale for CO2 fluxes. And another thing I want to point is that for all of the paired sites, the dissimilarity of land cover was always below 50%. But you might already notice there are two sites with similar land cover but different EVI. The same for the tundra sites at Naval Ridge is that we notice AmeriFlux tower is more rocky for the tower surroundings. And the situation for Sand Radar is we are comparing data from Lockheed Hill to NEON Sand Radar study sites. The difference in mean values for EVI was small, but they are just a significant difference. To wrap up, the main takeaway is for most paired sites, the pattern of annual variations were different. For most site years, the annual carbon balance for NEON and flux appears to be different by 50 grams of carbon per square meters per year. That means the agreement at half-hourly scale does not translate into good agreement at annual scale once gap filling is done. We have to recognize that a single tower may not adequately capture broader ecosystems. Thank you.
Fascinating. That was so interesting, Yujie. Thank you for sharing that work with us. That is really surprising and thought-provoking to see those differences. So thank you so much. I'm just going to check the Q&A, not necessarily seeing a question right now for Yujie. Does anyone want to throw one up quickly, or we can move to our last speaker and have general Q&A? Okay, Yujie, let's move on. We'll hear from Kit, and then again, all the talks that you've heard today, any questions for any of our speakers, you can just start populating them in the Q&A. We should have at least 10 minutes at the end to have a good discussion. So our last ambassador today is Kit Lewers. Kit is a PhD student at the University of Colorado and a NASA Finest Fellow who studies how information overload impacts institutions grounded in big data, especially how it relates to interoperability across fields. She develops modular, event-driven microservices architecture, which is also known as EDMSA, to integrate and process complex multi-source remote sensing data sets from NASA's airborne and spaceborne instruments, such as G-LIGHT, Everest-NG, and the upcoming Surface Biology and Geology mission, or SBG. She's also building workflows to explore cross-sensor interoperability and compatibility with major biodiversity initiatives, including NEON AOP. So thanks so much, Kit. Take it away.
Okay, cool. Can you guys see my screen?
Looks great.
Okay, cool. Hi, I'm Kit, like Samantha said. So my presentation is going to be a little bit different than some of the previous ones. Recently did a webinar using the Python Google Earth Engine API with the GBIF, or Global Biodiversity Information Facility API, to incorporate biodiversity records and AOP imagery. So this, I need to be able to advance forward. So this webinar happened in July, like I said, there were over 200 registrations worldwide, and there were two main tutorials that I went through. The first one was looking at Landsat imagery and calculating spectral indices and combining that with GBIF records. And then the second ones, which I will be talking about in this presentation, we're looking at AOP imagery with the NEON biorepository data to look at abundance and presence data, abundance data and then presence and absence data in the NEON biorepository, specifically a carabid beetle data set. And at the end of this presentation, I do have QR codes that you can scan to get the link to the full webinar and to the notebooks that I developed with this information in it. So I'm just going to quickly go over what the Google Earth Engine API is and what Google Earth Engine is in general. If you've never used it, it's really nice. There is a console version where you can just use directly in your browser, but it's in JavaScript syntax, and I would rather eat glass than use JavaScript on a daily basis. So I use the Python API. And it is a cloud-based geospatial analysis platform. You can access just petabytes and petabytes of satellite data, AOP data. And what's super nice is you can interact with that data and run scripts, automations, workflows across that API without having to have a bunch of data on your local machine, which is really valuable, especially with data like the NEON AOP data that's super high dimensionality and also just spatially very large. And it allows for data fusion and analysis once again. And the other cool thing is you can use multiple sensor platforms together if it is published on Google Earth Engine. And the hyperspectral data isn't the only NEON AOP data that you can access on Google Earth Engine. There's also RGB data. I'm not sure about the LiDAR data sets. That's not a question for me, but TLDR, you can get a lot of data and not have to use a lot of your own local resources to work with it, which furthers interoperability because then you can take even more big data from other places and learn about it. And speaking of those lovely APIs, the Global Biodiversity Information Facility, or GBIF as I call it, also has a Python API, and it does have an R API too if people are more inclined to use R. But as a facility, it is a global biodiversity data aggregator, and it mobilizes over 2.5 billion occurrence records. And I think that's actually closer to 3 billion now. You can check their website to confirm or deny that. And it is also very nicely laid out in standards-based infrastructure. So it's built on Darwin Core, which makes all of these different data repositories very interoperable with each other. So the Python API specifically enables programmatic access to species occurrence data, taxonomic backbone, data sets, and metadata. And the only thing that you really need to use it is a GBIF account, which is free. So it's great for supporting research applications, and once again, also great for integrating with other data sets very easily in just one notebook. So on to the analysis that I walked through. I am not an ecologist, so you'll probably look at this and be like, interesting choices. But this is just kind of a starter to start looking at this data and what you can do with it. And I really encourage y'all to look at the notebooks or look at the tutorials and like take it off and go run with it and make what I did way better. So what I decided to do was look at the Great Smoky Mountain region at the 2016 and the 2017 imagery, because in between those flights, there was actually a pretty big fire that happened in the region, and you can kind of see that right here. This very dark area is what I used to calculate the difference in burn, basically. So I took a normalized burn ratio of both years, and then I did a subtractive image and calculated pre versus post fire indices to identify that, and took it a step further. And this is another super nice thing about Google Earth Engine. I used a built-in method to classify pixels as burnt or not burnt. And it was just very simple vectorized function. So once I did all of that, I started bringing in a couple of sites, spatial sites, where I knew that there were beetle collections, and I linked that with the burn scar data. So I looked at the carabid beetle data, and the first analysis that I did was presence absence, because if you're simply looking at a like an event ID it's not a perfect indicator of abundance. For example, let's say that you got a beetle from a trap, and then from that trap, you also pinned that beetle, and then you took a little leg off of that beetle to have a like genetic record. One beetle could be showing up as 3 records in this. So this is, the first one is for present absence when I was doing the event ID, but you can also use specific NEON data sets to get abundance, which is what I did in the second analysis. And it's nice, you can either just pull everything down from an event ID from GBIF, or if you know your specific data set ID, you can pull everything down and run your analysis on that. And that's exactly what I did here. And the code also shows how to create interactive HTML exports, and they're super fast. You can drag and drop them. There's multiple layers. I think it should, yeah, there's multiple layers that you can look at and generate. It's super nice. And it also outputs GeoJSON. If for some reason you don't want to use EMAP or Google Earth Engine, you can also just generate data to put in your preferred package or spatial analysis software. And it also just showed how you can use these 2 data sets together. They weren't not designed to be used together, but they didn't go out and say, I specifically want Google Earth Engine and Python and the PyGBIF API to be usable together, but they are, and it's really nice, and it's a really fast and easy way to do exploratory data analysis with biodiversity records and geospatial data. So the results, there wasn't a super strong correlation between abundance from one year to the other, which is what this graph is, but there were a few species that were an outlier, and these are only 2 years, so probably not like the most robust data set that I could have used. And the other thing that I did when I mentioned doing this normalized burn difference ratio is this is a mask of all of the pixels that were considered to be burned. And I did a couple of analyses looking to see if there was any correlation between beetle abundance and how severe a area was around where they were burned. And I didn't see any correlation for that either. But once again, if you do want to watch the webinar, you can scan this QR code, or if you want to look at the NEON notebook, which also has all of the results from my other analyses that I used combining these 2 APIs, you can do that with these QR codes, and I can also drop a link in the chat in just a second. And yeah, if you have any questions, feel free to ask them here, or you can reach out to me at this email. And I'd be happy to talk about it. I absolutely love doing data analysis and helping people do data analysis. So feel free to reach out.
Fantastic. Wow, that's really exciting developments, Kit. Thank you. Yeah, that would be wonderful if you could drop some links in the chat once you're done presenting and able. Wow, what a fantastic set of talks. You all are working on really exciting and obviously very different aspects of NEON, which is super cool to see. So I think we're into the Q&A and discussion part of our talk. Please populate the Q&A with questions or discussion topics for the speakers. I think we'll go back. We'll start with the first question was asked of Reid, and he gave us a written answer. But Reid, if you could come and read the question that was asked to you earlier and expound on your answer, that would be great.
Yeah, so the question was, how well developed are the reference libraries for detecting some of these ultra small prokaryotes? And because there's been so many from these metagenome assemblies, I think there are some pretty decent references out there, but the genomes themselves are all super different from each other. So if there are some really unique genomes, it is definitely possible that they're still being missed. Especially a lot of the references are coming from environments like groundwater. So especially in the soil where these are maybe a little bit less well known, that it is possible that some are being missed. So we'll try to continue to develop those references as well.
I had a question for you too. I think you might have said it and I missed it, or maybe you could just reiterate, but do these ultra small organisms have a specific function? Do they tend to be, they do something specific in soil where you're finding them, or they're probably functionally extremely diverse, just like not ultra small things? Could you speak on that?
Yeah, so I think the ones that have been described so far are thought to be like parasites on other bacteria. So they might live on the surface of other bacteria and parasitize So it could be very important in things like carbon cycling and nutrient turnover. But I think it's exactly like you said, realistically, they're probably functionally diverse, and there's so many of them that it will be a while, I think, before that's fully uncovered.
Great. Thank you. All right, so the next question we had, I think, was for Alexi, and that was, did you run into any challenges with DNA quantity in the aerobiome samples? And if so, do you have any tips to share?
Yeah, so I didn't really actually run into any challenges getting enough DNA out of the Santa Rita samples. I guess one good thing about the desert is we have a lot of dust, which microbes like to cling to. But our larger international team has been sampling all over the world, and we do have environments where there are far fewer microbes in the air, so they've had a bit more trouble. And so what we've been doing is pooling multiple filters together to get more DNA out that way. And there should be kind of a methods paper coming out of our research group maybe in the next year or two, kind of providing some tips on filtering and DNA extractions and sequencing and things like that.
Awesome. And then I think there was another one for you. What is the mechanism of impact from air temperature on microbial communities? Is that the warmer air itself killing off certain sensitive microbes, or the warming on the ground is changing which microbes are being launched in the air in the first place? What do you think?
Yeah, so that's a really great question. It's something we're still discussing and thinking about. Given that we're sampling microbes only about 4 meters off the ground, I think that the temperature either influences something or correlates with something that's influencing which microbes are like aerosolized and brought into the air. And then once you get higher up in the atmosphere, we know microbes stay up there for much longer. I think temperature can really influence which microbes survive because it can get very cold.
Right. Very interesting. I guess I had a question for you too, since we're on your paper or your study. The fungi, that was really striking how it seemed like for soil or for bacteria, you could maybe point to a soil source for a lot of those, but fungi were so different. Do you have any speculative thoughts on where those atmospheric fungi are? They coming from plant leaves or where?
Yeah, that's a really great question. They could be coming from plant leaves, so like the phyllosphere. I mean, fungi are known to disperse through the air really well with like their spores and things. So it's possible that they could also be coming from more distant sources. That's definitely something that once we can get our source tracker analysis going, we might be able to tease apart a bit more if we have some different potential sources of fungi to look at.
Thank you. All right, Yujie, we have a question for you. Have you looked at other flux data comparisons between sites? So maybe not just like CO2, but the water fluxes or maybe energy balance or anything else comparing the two towers?
Yes, they did. That's a really good question. I didn't present results for sensible heat and latent heat fluxes in this presentation. For your message, the agreement for sensible heat fluxes was generally better than that for CO2 fluxes and water fluxes. And the pattern for like agreement for water fluxes were different from what I presented for CO2 fluxes. For example, I mean, the sites I pointed with good agreement for CO2 fluxes were like Harvard Forest, Burled Forest, Haley, but for water fluxes, the sites with good agreement are Harvard Forest is still here, Rocky Mountain and Yodi. So that means the level of agreement are different for different measured fluxes. That's something, a big thing for this project, because we realize there are too many variables we are trying to compare. Also, the meteorological variables, I also didn't show it in the talk. So the plan is to come up with scorecard combining different variables and trying to assign points depends on different correlation metrics, and then give us score as a summary for different sites for the level of agreement. That's something working in progress. Thank you for the question.
We did have another question come in for Alexi. Alexi, I know you answered it written, but why don't we go ahead and go through that? So it was, have you observed any correlations between soil types and microbial communities in the atmosphere?
Yeah, great question. And this is something that we're just starting to look at. So both at Santa Rita, as I mentioned, I'm starting to investigate that. But again, also our larger team of researchers for all the air samples we're getting across the globe, will also taking underlying soil samples. So I definitely think that different soil types will have different like aerosolization potentials, which could strongly influence which microbes are emitted into the atmosphere. So that's something that we're excited to look into a bit more.
Very cool. We have a couple more minutes. We'll just give it some time, see if anything else pops up in the Q&A. Or like I said, if anyone would like to, if you do raise your hand, we can give you permission to ask a question over audio if you don't like typing. Well, I see that Natalie has a question. Are we able to give Natalie permission to unmute?
Go ahead, Natalie.
Oh, okay. Sorry, I thought this might be faster than typing. I'm just wondering with the microbial study, since the functional pathways are really sort of hypothetical at this point, like what is the management, like what's the ultimate outcome of the study of understanding the outcomes of the correlations between atmospheric microbes and presence? Do you know what I mean?
I think that one was for you, Alexi.
Okay, so sorry, can you repeat that?
Oh, can you hear me still?
Yeah.
Gosh, good thing I didn't know I was still unmuted. Okay, I was just wondering kind of what's the sort of management implications of your study? Like, since the functional pathways of microbes from our RNA is still pretty hypothetical, where are you trying to go with it, basically?
Yeah, I think the kind of the broader goal, like I mentioned at the beginning, is just to start thinking about the atmosphere as a potential ecosystem and not just this passive mechanism by which microbes move from like one, land surface to another, investigating if they're active while they're in the air. And so understanding like, do they have genes that, and again, like you said, it's still an imperfect science, that might help them be adapted to the harsh life in the atmosphere, is just a way to start looking at that. But this is definitely early stages because it's hard to get enough DNA out of these samples. And then when you do sequence, if you have a lot of like eukaryotic DNA, it can really drown out those more like bacterial and archaeal megs that we want to look at.
Awesome, thank you. Let's sneak in one more question for Yujie, but I did put in the chat, we have another NEON Science Seminar coming up in a month. It's October 7th, and that will be Tanya Berger-Wolf will be speaking about AI for nature, from science to impact. So hopefully that'll be a great talk, and we'd love to see you there. Yujie, can you speak about, with considerable differences in the footprints between several AmeriFlux and NEON sites, is data integration between these sites still a viable option?
Yeah, that's a great question. That's a research question we want to come back. How to say from the findings? You need to be very, very cautious when integrating the two data streams. There are also many other reasons, like the sensors are different and the measurement protocols and data QA/QC are all different for NEON and AmeriFlux measurements. Currently, we have 7 years of data overlapping. We already observed the big differences in the measurements. So my conclusion is you need to be cautious for data integration.
It's really a great point that the Niwot Ridge AmeriFlux site and the NEON Ridge site are not necessarily the same thing. It's really a very important point, Yujie. Thank you for bringing that to all of us. So yeah, I think we're out of time. Wow, what a fantastic set of talks. Thank you so much to ambassadors for sharing your research, what you're up to. We're excited for their participation in the NEON Convergence Summit this fall. And thanks again. We do have another seminar coming up October 7th. We'd love to see you. And other than that, have a great rest of your day, and thanks for coming.